Gunjan Sharma to introduce our friend Dr. Poonam Mishra to today's audience. Dr. Gunjan, please.
Thank you, sir. It's my pleasure to introduce Dr. Poonam Mishra. She did her master's in biotechnology from BHU Varanasi and then completed her PhD in yeast developmental genetics lab under guidance of Dr. Jagmohan Singh at Intech, Chandigarh, to find out the occurrence of prion-like behavior of CUT4 protein, a constituent of telomerase promoting complex, thereby regulating cell cycle in fish and yeast. In the meantime, she also identified a method to increase the recombination efficiency for the tagging and deletion of genes in fish and yeast. Her expertise is in yeast genomics and proteomics. Further, she joined Department of Microbiology and Biotechnology as an assistant professor at the MSU Baroda and continued for 4 academic sessions. Last year, she has joined the Department of Biotechnology at Dr. Kirana and Pallavi Patel Global University, Vadodara. Welcome, ma'am.
Thank you so much, Gunjan. Thank you
Dr.
so much.
Dr. Poonam, as a small request, please hide that notification about sharing of the screen. Because
Okay.
Thank you so much. Please continue.
Is it visible at your end, sir?
Yes, it is visible. Yes.
Okay. Thank you, Gunjan, and thank you, Dr. Shinai, for providing me this opportunity to actually speak as a speaker, to speak in this World Fungus Day 2023 lecture series. So, why I have chosen this topic, fungal phytopathogens, alternative biocontrol agents, because recently when we have started looking for some kind of research activities and using my own expertise in the field of yeast genomics, then we thought of looking for certain kind of problems that are actually on the real time, which are being faced by the farmers and the farmers at the different levels. So one among them is basically the attack by the different phytopathogens or which are belonging to the different categories. They are causing the damage to the crop. So why it is essential for us to look for the alternate methods by which these can be controlled, because it can be, as you know, that presently itself we are present in a large number on this planet, and we are expected to increase up to 10 billion people on the planet by the 2050. By the year 2050, we are supposed to be 10 billion. And one of the biggest challenge that would be faced by any of the country, whether it is developing or it's a developed country, to feed all those population. And why I'm saying that feeding them, because despite of having minimal agriculture production at every country, there is a loss of crop production or overall yield of the plants, crops, because of the different diseases which are caused by the phytopathogens. So there results in the loss of around 10 to 50% of overall productivity, which is being done by the crops or which are being carried out by the different, you can say, the different grain products. So all of them, they are causing the reduction in overall plant crops because of certain these diseases. So what are the common different kind of phytopathogens which are known to produce these damages? They are because of Alternaria, Aspergillus, Botrytis, Cladosporium, and so many so to cite on. So why we need these agents or why we need them? So are there certain options which were available for the people to use in these to avoid these agricultural damage? So from there, the idea, we are very well aware of one of the very popular scientists, Professor M.S. Swaminathan, who just passed away 2 days back. He was the father of Green Revolution in 1960. He proposed the better living through the chemistry by the uses of different chemical pesticides and the fertilizers, so as to increase the crop yield and feed this to the population or the human population across the world. By it, or if we are talking about the Indian perspective, then feed them to the people of the India. But what was the you can say what was the after effect of these chemical pesticides and the fertilizers which are being used to the population? So these can be on the basis of the detrimental effects on the environment, which could be loss of biodiversity, reduction in the soil fertility, disturbance in the quantity and quality of the microorganism, and also the pollutant related toxicity which was induced by these chemical pesticides and fertilizers when these were used in a excess, so as to increase just the crop yield. Whereas they are also going to affect the human health. Why? Because of the occupational poisoning, because the farmers who are using these pesticides, they are getting exposed to different kinds of chemical substances. This is leading to the childhood obesity, childhood diabetes, and also they are affecting the intellectuals of the child and leading them to the IQ loss and as well as to the intellectual disabilities. So overall, if we say that the uses of these chemical pesticides and fertilizers was not so much providing the good to the human population, whether it is for the developing country or for the developed country. So what could be the alternate possibilities which are available for the scientists to go on with? So that was the use of the biocontrol. Now, if we talk about the definition or the basic definition of the biocontrol, so just to say it, or just to what it indicates from the term itself, that it's a use of action of parasites, predators of the microorganism in maintaining the lower population density of the pathogenic organism, which otherwise would not be possible. And this could be based on the target of suppression of which kind of pathogenic strain that they are using, the number, type, and the source of biological agent, as well as the degree and the timing of the interventions to the different biological processes of the parasite that are causing the infection to the plant, and thereby reducing their overall crop yield. So if we are talking about the biocontrol, then in a larger scale, there are different kinds of biocontrols which are available in the market, which are available belonging to the different kinds of categories. This may include the nematodes, this may include the insects, but what we are focusing for the today's lecture, or what we are trying to get into it, that is because of the microbial cells which are belonging to the bacterial, viral, or the fungal kind of biocontrol agents that are major focus of this talk. Now, how these may function? They may function by two ways. They can be biostimulant or they can be biopesticides. When I'm talking about the biostimulants as the microbial cells, what they are actually doing, they are causing the help in the improvement of plant health by itself through the plant extract or the natural compound, thereby increasing their own activity against the different kinds of pests so that they can survive on their own. Whereas when I'm saying about the uses of these microbial cells as the biopesticides, and what I am actually trying to say, it is for the inhibition of the pathogen growth in the plants itself. So thereby they are causing the damage to the pathogens, and then if the damage to the pathogen is there, then they are not going to affect the plant by themselves. So if we just want to summarize the use of this word biological control, then there are three major partners. One is the living agent, which is the pathogen. The other one is the pest, other one is the biocontrol agent, and the third one, which is the most important, it should be directly or indirectly for the human good. So since we are very much, we already studied, or we are reading about, or we are talking about the host pathogen interaction or the uses of different agents against the pathogen. So until unless they are not used for the good to the human, to the crops which are doing good for the human benefit, so until unless those terms cannot be utilized for the biological control. So if we talk about the distribution of these biological agent or these biocontrol agents, then we can distribute, or we can distinguish them based on the different categories or different criteria. If we talk about based on their application, then they can be either biofertilizer or the biocontrol agent. If we are talking about the overall content of those organisms which are making these biocontrol agents, so these can be bacterial cells, these can be fungi cell, and sometimes the consortia of these bacterial and fungi cells in the combination, they can also be used as to function for the biocontrol agent. And if we just look for the different distinct types of the microbial cells which are making or which are forming these biocontrol agents, so these can be belonging to rhizobial cells, entomopathogens, mycorrhiza, as we have earlier also seen in the previous lecture by the Dr. Bhagyaraja, that there are certain mycorrhizal cells which are functioning as the biological controls, and also they can be the mix of the epiphytic fungi, soil fungi, or there could be through the PGPR. So we can just distinguish them, and we can just classify them based on the different types of properties on these different categories. Now, from where all these microbiota, or from where all these fungal cells that we are saying that they are functioning as the biological agents, they are coming from. They are basically coming from the overall phytobiome of the plant itself. So if you are saying that phytobiome, then they can be distinguished into certain kind of categories. The first one is the philosphere, which is the complete microbiome from the part of the plant which is just above the ground portion of the plant, or the ground portion above the ground portion, that is known as this philosphere. Then we have this rhizosphere, which is the area surrounding the root region of the plant. So the microbes which are residing there, or the fungal cells which are residing there in the nearby places of the roots of the plants, they are having these microbiomes which can be potentially function as the biocontrol agent when tested for the certain against the certain kind of microbial cells. And apart from that, we can have the endospheres, which are the microbiome cells or the microbial cells which are existing inside the internal compartments of these plant cells, of these plants. So that now why I am focusing on this slide, because all the microbiota or the fungal cells or the microbial cells, what I am trying to say, they are just in the nearby places to the plant itself. They are going to provide certain kind of resistance against the infection, resistance against the pathogen, either by the different kind of mechanism that would be there. So if we just have an idea that what kind of the biocontrol agents are there, they can be in the form of microbial cells, in the bacterial cells. So these are the known agents, or these are the known bacterial agents: Azospirillum, Azotobacter, Bacillus, Pseudomonas, Rhizobium. These are all the bacterial cells. If you just focus on these bacterial cells, they are having their specific function against the target pathogens. So they are inhibiting the growth of those pathogens. They are increasing the effect on the test plant or the disease plant onto which these target pathogens are functioning on. So in the same way, if we look for the fungal biocontrol agents, now we have so many reports as being suggested by this tabulated form that there are certain fungal strains like the Aspergillus, there are Trichoderma species, Penicillium, which is one of the very first known kind of fungal strain to have certain kind of the antibiotic kind of activity. So if you see that this fungal strain list is dominated by the Trichoderma, and it has the it can provide the immunity against various kind of pathogens which are belonging to the different kind of fungal strains, like the Rhizoctonia, like the Bipolaris, like the Sclerotorium, and also they are providing the immunity to the different test plants against these target pathogens to causing the disease to these plants. So what could be the different kind of mechanism which are being utilized by the biocontrol agent that can be carried out or that can be used by these fungal cells or the fungal phytopathogen to provide certain kind of immunity to the plant against those target pathogen? So these are some of the very common known mechanisms which are being shown here. The first one is the competition. When I'm saying about the competition, so this competition is between the phycopathogen and between the biocontrol agent. For what? It is for the space, it is for the nutrients, so that because we know that the area or the rhizosphere, if we just focus on the rhizosphere region, then it may be having the limited source of the nutrient as well as for the space. So if there is a competition for the nutrient and space, then one of them, or among the consortium, one of them will be able to outgrow the other kind of or the other fungal or bacterial strain. So thereby they are going to affect the growth of those strains which will not be able to survive in those competitive conditions. So the first way or the one way is the competition. Second way is the antibiosis, where the certain kind of secondary metabolites. These are basically the metabolites or the volatile compounds which are being secreted by these biocontrol agents, which are going to infect, which are going to cause the retardation in the growth of those phycopathogens so that they may not survive in those conditions. So the production of certain chemical compounds. Then the induction of this plant resistance, that is also known as ISR. So what does they do? They help in the they actually help in the secretion of certain kind of plant hormones like the gibberellic acid or the thylline that can induce certain specific responses in the plant, and that can lead to the accumulation of certain favorable microbialites, so microbial substances which can help in the help in providing the immunity to these plants against the certain kind of pathogens. And apart from that, one major mechanism which is mycoparasitism, in what the fungal cell they do, they directly attack and the parasitize the pathogens, and this predatory behavior under the nutrient limiting condition, it helps in the microbial predation and thereby killing of those phytopathogens by these biocontrol agents. So if we just want to summarize all these mechanisms or the other possible availabilities or the available possibilities by which the fungal phytopathogens, they can just inhibit the growth of those kind of can be inhibited by the biocontrol agents. So that could be the direct antagonism, as we have seen, by the mycoparasitism or the hyperparasitism, by the secretion of certain non-lytic mycoviruses, by the different other biocontrol agents like the trichoderma. Then we have this mixed path antagonism by the different mechanism, antibiotic secretion of the lytic enzymes, unregulated waste product, physical and chemical interferences, by the different other secondary metabolites which are being secreted by these biocontrol agents, and also the uses of this indirect antagonism by the use of this competition and induction of host resistance, whereby there is the direct contact with the fungal cell wall and it can initiate the pathogen associated molecular patterns and thereby inducing the resistance in the plant itself, so that the plant can resist the attack by the phytopathogens. So these are certain some of the mechanism by which the fungal phytopathogens or these biocontrol agents, they inhibit the infections of the other fungal phytopathogens. So what are the different kinds of mechanism by these fungal biocontrol agents? As I mentioned itself, that there are the release of various volatile, non-volatile antifungal compounds which are being produced by the fungal biocontrol agent. These could be, some of the examples are gliotoxin, gliovirin, which are being very much secreted by the trichoderma, which is very known or well-known potent kind of biocontrol agent which are being used, or the fungal cell which is being used as a biocontrol agent. It helps in the overall plant growth development by the developmental characteristic. It inhibits the mycelial growth. It causes the suppression of the fungal pathogen, maintains the soil fertility, increases the beneficial microorganism, and also works in a eco-friendly manner. So that these are now becoming a choice of action or choice of more uses by the government, by the scientists, and by the farmers at the field level, so that they can enhance the crop yield, they can enhance the crop productivity. So this is again a kind of example that how these fungal biocontrol agent, they can have the different mechanism. If you see that they can have the locust biological control by just laying off the fungal spores on these locusts, by which they are causing the degradation of the cell wall of these locusts, thereby causing their death. They can have the parasitism, antibiosis, competition among the different other phytopathogens. They can dissolve the cell walls of the pathogenic fungi. They can have the toxins, which are actually the secondary metabolites, which are released in the nearby region so as to have the herbicidal activity. So in and all, what I want to try to convey you, that the role of these phytopathogenic fungi as a biocontrol agent, they can have the different kind of mechanism for the action against the different phytopathogens. They can be used, and they can be used as a function for the biological control. Now, if we are talking about the fungal biocontrol agent repeatedly, then what are the different methods, or what are the laboratory methods which are used for the identification and screening of these fungal biocontrol agents? So it's a very simple, if this picture actually shows you that this is a very kind of simple experiment, but it's not like that, because for that every time you need to have certain standardized condition to be followed up, so that you can have the actual access of the function of these fungal strains to function as a biocontrol agent. So just the simplest one is the dual culture, where you just put these putative fungal biocontrol agent, as well as to the pathogenic strain on the same kind of agar media. If you get the resistance, if you get such kind of observation that the inhibition of the growth of these fungal phytopathogen is occurring, then this is being functioning as a biocontrol agent. If you are supposing that the fungal strain, it is producing certain kind of volatile biocontrol compound, then you can put them in a kind of chamber kind of assembly, or the petri plate, closed petri plate kind of assembly, and where you can have the observation for the growth of these bacterial cell, or sorry, of the growth of these pathogens against those fungal phytopathogens. And similarly, this can be repeated into the liquid medium or directly onto the different plant parts. They can be tested for the antifungal activity if they are anti-phytopathic activity, if they are producing any kind of effect there or not. So this could be done. So if this is the scenario that these can be screened, so why it is now the situation that why these fungal pathogens or the biocontrol agents are not very much into the concept, not very much popular phenomena as just of those fungicides or the pesticides that were very frequently used by our farmers. So why it is so? Because if you see through, then this process of commercializing fungal biocontrol agent, it's a kind of stepwise production validation which requires a long process. First of all, as I said, that there should be the microbial screening. I have just tried to show you in a very simple way, but this process, it takes place certain specific time of 6 months to 1 year, depending on the conditions of the labs or how you are performing that. So starting from the microbial screening, then you go for the characterization part for checking up the potential of these fungal strain to function as a biocontrol agent. Then you have to carry out the formulation for those chemical or the secondary metabolite which are taking up from these fungal cells. Then once you get that, you have to protect it by the law through the patents, and then you have to register it. And this complete process may take a large kind of it's a time-taking process. So no doubt, people are now shifting towards the biocontrol agents. People are shifting or looking for more safer options for their crop yield, increase in the crop yield and everything. But to the actual level to function it like that, it may take some time, some more time and more research also. Despite of that, if you see that there are certain commercialized fungal biocontrol agents which are known to exist or which are known to be functioning at the commercial level. So if you see that these are maximally, you can see the production is carried out by the developed country, and it is not very much prevalent in our developing country or some other countries also. So you can see that there are the certain products like AQ10, like the Primastop, like the BNAPT. These are all containing one or other kind of biocontrol agent which is from the fungal phytopathogen. So they themselves are providing certain kind of immunity to the plants against the target pathogen through the help of these commercialized fungal biocontrol agents. So what is the future perspectives or the opportunities associated with these biocontrol agents? So here you can see that at present, if you see that the overall worldwide market for these biocontrol as a fungal biocontrol agents, it is around $4,800 million of the conditions or the revenue can be generated, but it is expected to be growing threefold in next 10 years. So if we consider it, then we can say that there is a lot of opportunities which are available for the scientists, for the world to explore in this field of the biocontrol agent, where we can use the fungal phytopathogens to function as an biocontrol agent. And there are different commercial partners that can be available to get into it. And in India, if we look for, then there is a company which is known as UPL, that is a kind of major player across the worldwide for the biocontrol agent production. So with this, I'll be thanking to the MycoIndia Journal for letting me have this opportunity to speak in this guest lecture series. So thank you, Shinoy sir, and thank you so much.
Thank you, Dr. Poonam Mishra. It was a very wonderful presentation. We'll take a couple of questions. Mr. Arvind, your first question.
Thanks for the opportunity again, sir. My first question is like, as you said, the BCA has a multiple role. Can it eradicate or reduce the weed which is being grown in the which is being grown along with the crop?
Yes, sir. Actually, there I have shown you a slide where they are showing the herbicidal functions also. So they may be tested for those herbicides and they can function as herbicides and weedicides also. But if we are saying for the function of these fungicides or these biocontrol agents to be function against the weed, then for that we need to check for the specific function. So whether it is functioning or not. So for that we need to check it.
Okay, fine. Thanks, ma'am.
Mr. Subhranshu.
Thank you, sir. Good afternoon, everyone. Madam, thanks for a nice presentation, actually. And this is actually my core interest is in biocontrol science. But as you said, madam, that there are so many biocontrol products available worldwide, but in India only 2 companies are producing.
Yes.
And also there are a number of biocontrol experiments are carried out all over India. But why? What do you think? Why we are falling short of formulating developing a formulation so that the farmers can use it? Why we are falling short in this regard?
Actually, if I say in my opinion, then as far as since we are belonging to a developing country, the major concern for us is for us or as a farmer, the low cost and how much the farmers are aware of these kind of conditions or the biological agents they are aware of. Major is the general awareness, low cost of production. That are the 2 major features that I think that the biocontrol agents, they are lacking as of now. Because chemical fertilizers, pesticides, these are very well known to the farmers are aware of them. So it is on us as scientists or on us as a scientific community to make them aware of the available options which are prevailing in the market itself. So that is what I am thinking that we are lagging. The general awareness.
That's one part is right, madam. But don't you think that getting registered a microbial product in India is not in the law, and even if the product is also to get the register, the product also is very difficult. Because myself, when I was doing my PhD in ICR National Rice Research Institute in Odisha, Kotak, I have also applied for a bacteria which was controlling the rice pathogens, but after it is being stocked in the law channel for 1.5 years, then after it was rejected. So don't you think that is also a hindrance for developing such biocontrol or biopesticide or those products?
Yeah, that can be one reason, because feasibility of getting it to commercialized manner, the product has to be converted into a formulation in a commercialized form that can be available to the farmers. That is one of the reason, or that is one of the reason for not getting the low cost product of these formulation, because if there is no competition